Welcome to part 2 of the voiceover lecture for Chapter 7, the nervous system. If you haven't yet, please watch part 1 of the nervous system, and then continue. Again, I've divided this chapter into 6 sections. We've already covered sections 1 and 2, an overview of the nervous system, and the nervous system in terms of its tissue. In this video, I'll cover section 3, the action potential. Subsequent sections will be covered in future videos. Section 3, Action Potentials. Recall from Chapter 3 the concept of cellular transport. In that chapter, we talked about how a resting cell has different concentrations of molecules and ions on the inside versus the outside of the cell. In other words, a resting cell maintains an electrochemical gradient. Outside of the cell, also referred to as the extracellular space, there are more sodium and chloride ions. Inside the cell, there are more potassium ions. Also, overall, the inside of the cell is more negatively charged compared to the outside of the cell. A charge separation is measured in terms of voltages. The average resting neuron has a voltage of -70 millivolts inside of the cell. And remember, this is referred to as a resting membrane potential. Do you remember what component of the cell helps maintain the resting state? Hopefully you remembered that a cell maintains rest using the sodium-potassium pump. Remember, this was a transport protein in the membrane of our cells that pumps 3 sodiums out of the cell and pumps 2 potassiums inside of the cell. Now, normally there's more sodiums outside and more potassiums in, and so the molecules are moving against the concentration gradient. They are moving against the normal flow of diffusion, and so this process is going to require energy in the form of ATP. So this was an example of active transport. Recall that the resting membrane potential of a cell measures about -70 millivolts. We use a device like the one pictured here to measure the resting membrane potential of a cell. This device is called a voltmeter. There's a probe that's inserted inside the cell. There's also an electrode that remains outside of the cell. And between those 2 electrodes, the device measures the charge difference in those 2 different areas. Remember, not all transport in and out of a cell is active transport. There is also passive transport, which does not require energy, but requires molecules to move with the normal flow of diffusion. Remember, that's the flow from a high concentration to a low concentration. One example of passive transport was facilitated transport. This is where molecules move through channels. Ion channels were protein channels in the membrane of a cell that allow ions to flow in and out of a cell with diffusion. Some ion channels are just like an open hole where the ions can move freely in or out of the cell. But some channels are gated. They can open or close in response to some sort of stimuli. One stimuli that can open or close an ion channel is a change in voltage. That is a change in charge differential across the membrane. These are called voltage-gated ion channels. Another type of channel is a chemically gated ion channel. These channels require a molecule to physically bind to the channel to open or close the gate. They are also sometimes called ligand-gated ion channels. The channel pictured here is a chemically gated ion channel. You can see on the left the channel is closed because there's no chemical or molecule bound to it. But the channel on the right is open because the molecule has bound to it, and now the ions can flow with diffusion through the chemically gated ion channel. Now I'd like to discuss an overview of the process of an action potential. In subsequent slides, we'll go over more details of each of these steps. An action potential begins in a resting neuron, where something opens a few sodium channels in an axon hillock. Recall that an axon hillock was the region where the axon meets the soma or cell body of a neuron. So a few of these ion channels are opened up. Because sodium's on a high concentration outside the cell, sodium's going to flow into the cell by diffusion. This influx of positive sodium ions is going to change the voltage inside the cell. The inside of the cell is negative 70 millivolts on average. An influx of positive ions will make the inside less negative. It's changing the voltage. A change in voltage will trigger voltage-gated sodium channels to open. These ion channels allow even more sodium to rush in, and more voltage-gated sodium channels will open even further down the axon. That's kind of the overview of the process of an action potential. Action potentials have 4 stages. In stage 1, the process of depolarization occurs. Stage 2 is repolarization. Stage 3, hyperpolarization. And step 4 is the return to rest, which I nickname the restoration stage. An action potential begins with the process of depolarization. Remember that the inside of a resting cell is negatively charged, about negative 70 millivolts. Some stimulus is going to open some sodium channels at the initial segment of a neuron. Remember, the initial segment was the area of the axon hillock where an action potential begins. So this sodium is going to rush into the cell, and it's going to reduce the difference in charge between the inside and the outside of the cell. Sodium's positively charged, the resting cell has a negative interior. As those positives move in, the inside of the cell is going to become less and less negative. In other words, it's becoming depolarized. Eventually, a certain threshold is reached. That means the inside becomes less and less negative to a point where voltage-gated sodium channels are triggered to open up. Remember, these are ion channels that allow sodium to flow into the cell, but they're normally closed unless there's a change in the resting voltage. The threshold that must be reached is negative 55 millivolts. That means enough sodium has to rush into the cell that the inside goes from a negative 70 millivolts up to a negative 55 millivolts. And now these voltage-gated sodium channels open up and much more sodium can rush in. This process is called an all-or-nothing process because either the channels are open or they're closed. They don't just allow sodium to trickle in at any point. Again, some stimulus has allowed sodium to rush in the initial segment of an axon. That sodium changes the voltage inside the resting cell. That change in voltage causes voltage-gated sodium channels to open, allowing even more sodium to flow in. All along the length of the axon are many voltage-gated sodium channels. So as sodium rushes in at the initial segment of an axon, that sodium is going to move down the length of the axon, changing the voltage in that particular area of the axon, triggering more voltage-gated sodium channels to open, allowing even more sodium to rush in. These voltage-gated sodium channels keep opening, more and more sodium rushes in, and this process occurs all down the length of the axon. Enough sodium rushes into the axon to the point where the peak membrane potential of the inside of the cell becomes a positive charge. It actually becomes about positive 30 millivolts. Whenever you have a flow of charged particles, whether it's protons, electrons, or sodium ions or chloride ions, a flow of a charged particle is called an electrical current. So in other words, the process of an action potential produces an electrical current that flows down the axon of a neuron. This diagram is depicting the process of depolarization that we just discussed. So here's a neuron, and we're going to zoom in on the axon of this neuron over here. So this kind of cutaway of a tube is a cutaway of the axon of that cell. On the membrane of this axon, sodium has begun to rush into the axon, causing the inside to become positive. As the sodium rushes in and makes the inside positive, voltage-gated sodium channels open, allowing even more sodium to rush in. Down below you can see a graph that depicts this change in voltage in each subsequent area of the axon. So way to the far right, nothing has happened yet, and so the voltage is still at rest, negative 70 millivolts. But over here near the initial segment, sodium's rushed in, and you can see the voltage measured in millivolts here on the y-axis. The voltage has increased, become less and less negative as positive sodiums rush in. This red line represents the depolarization process. The dashed line here at about negative 55 millivolts is the threshold where voltage-gated sodium channels will open. The next stage of the action potential is termed repolarization. You might be able to guess from the name that this is going to be when the inside of the cell returns to being negative. So we said that sodium ions have begun rushing into the cell, making it become more and more positive. The inside of the cell will reach a peak voltage of about positive 30 millivolts. So the inside does not become any more positive than that usually. At this point, those voltage-gated sodium channels are triggered to close. And another type of channel will open in response. These channels are called voltage-gated potassium channels. So when they open, they allow potassium to flow across the membrane. Remember, potassium is found in a high concentration inside the cell. So by diffusion, potassium wants to leave, wants to move out of the cell. So when these channels open, potassium flows out. Potassium has a positive charge. If positive charges are leaving the cell, the inside is going to start to become more and more negative. In other words, the inside membrane potential will decrease as potassium leaves the cell. This diagram is a continuation of the previous diagram, showing the process of an action potential. You can see in that initial segment that potassium has now begun to rush out of the cell. Seen here, potassium is leaving the cell. Remember, potassium is positively charged, and as those positives leave, the inside becomes more negative. They can also be seen below, depicted in the graph. The inside of the cell has reached a peak of about positive 30 millivolts, and as potassium leaves, that voltage drops. It goes down and down and down, back down towards negative 70 millivolts. Now, in the areas where the process is just starting to occur, sodium is still rushing in on this part of the axon, and so the inside is still becoming more positive. Further down the axon, a little bit of sodium has kind of diffused over to the right, and so you can see a little bit of positivity or a little bit of a less negative voltage in that area. And then even further to the right, nothing yet has happened, and so the cell is still at its resting membrane potential. Let's review. So far we've talked about two different types of ion channels: voltage-gated sodium channels and voltage-gated potassium channels. Recall that voltage-gated sodium channels open once the threshold of negative 55 millivolts is reached, and they close at a positive 30 millivolts. At that time, at that positive 30 millivolts, the voltage-gated potassium channels will open. Now, what causes potassium channels to close is actually a delayed timing effect. So those potassium channels close, but they close fairly slowly, and they actually allow extra potassium to leave the cell. And this creates our next stage, hyperpolarization. You can kind of guess from the name that the inside's going to become even more polarized, even more negative than it was at rest. Again, voltage-gated potassium channels are fairly slow to close, and they allow an extra amount of potassium to leave the inside of the cell. That causes the inside to become even more negative than it was at rest. Recall that rest was about negative 70 millivolts. So in hyperpolarization, the inside becomes about negative 80 millivolts. This process helps to prevent action potentials from firing too quickly and is known as the refractory period. This process also ensures that action potentials only occur in one direction. In other words, an action potential is unidirectional. We always want the signal to travel from the cell body of a neuron down towards the axon terminals. Finally, we have the last stage, restoration. So we've had the sodium channels close, the potassium channels have closed, so there's no more diffusion of ions across the membrane. But during this process, and at all times, the sodium-potassium pump is always working. It's always pumping sodium and potassium ions. The sodium-potassium pump is much slower than ion channels are, but once those ion channels have closed, the sodium-potassium pump now has a chance to restore the neuron back to its resting state. That is, it's going to pump those sodiums back out of the cell, and it's going to pump the potassiums back in. And it's going to cause the inside of the cell to return back to its normal resting membrane potential, negative 70 millivolts. In this diagram of the axon, you can see more time has passed, and that initial segment where the action potential had first occurred has now gone back to rest. So the sodium-potassium pumps have kicked the sodium back out and potassium back in. So the inside's back to negative 70 millivolts. Now in this region of the axon, we're still at the stage where potassium is flowing out of the cell. The cell is becoming hyperpolarized as extra potassium leaves. In this region of the axon, sodium is still rushing in. So we're kind of at one of the first stages, the depolarization stage in this region of the axon. As that sodium is flowing in, some of it's going to move or diffuse to the right. And so this region here to the right has a bit of a positive change. So it's gone from negative 70, it's almost to negative 55. So threshold has not yet been reached. And then finally, even further to the right, nothing yet has happened. So the cell's still at rest. You can kind of see from this process how an action potential will always travel down the axon from the initial segment outward and towards the axon terminal. This diagram represents the graph that we've been showing in those previous slides, but it's complete. It's showing the whole process of an action potential in one region or one patch of an axon. Again, on the x-axis is millivolts, and on the y-axis is time, measured in milliseconds. An action potential begins when the cell's at rest initially at about negative 70 millivolts. Something's going to trigger some sodium channels to open, and sodium will rush in, and the inside starts to become positive. If enough sodium rushes in, we reach threshold, which here it's showing about a negative 50 millivolts. Some texts say it's negative 55. It varies from source to source. I'm going to go with the negative 55 number though. So at about negative 55 millivolts, enough sodium is rushed in, threshold is reached, and now voltage-gated sodium channels pop open, and they allow a huge influx of sodium to rush in the cell, and the inside becomes more and more and more positive, and peaks at about positive 30 millivolts. So this region here of the graph in red, this is the depolarization process. At about positive 30 millivolts, those sodium channels snap shut, and voltage-gated potassium channels pop open. This allows potassium to flow out of the cell. In other words, the inside becomes less positive. So you can see that the voltage inside of the cell begins to drop in this blue line here. That's the process of repolarization. Remember, potassium channels are slow to close and they'll allow an extra amount of potassium to leave, and so the voltage inside the cell dips below the resting state. So this kind of curved part of the line here is known as hyperpolarization. It's even more polarized than it once was at the beginning. Finally, the sodium-potassium pumps are able to do their work, and they return everything back to rest, negative 70 millivolts. So this part of the line is the resting stage. This is an animation depicting an action potential. This horizontal line is the membrane of a neuron, and in that membrane you have your two types of channels, the sodium and potassium channels. The sodium channel opens, allows sodium to flow in, and the inside becomes positive. The potassium channel opens, allows potassium to leave, and the inside becomes negative. I'd watch this animation several times, maybe describe it to yourself, and ensure that you understand this process of an action potential. If you click on this picture, it's a link that'll take you to YouTube, and you can watch a video describing the process of an action potential. Action potentials are fast, very fast. You imagine we'd want them to be fast because when you want to move a muscle, you want it to occur quickly. In fact, action potentials are so fast, they are faster than the fastest production car. I think it's called the Bugatti Veyron Supersport, which broke the world record of a top speed of 268 miles per hour. Action potentials are faster. An action potential travels down one section of an axon in 2 to 3 milliseconds. That's about 130 meters per second or 290 miles per hour. Action potentials can repeat several hundred times per second. That's including with the refractory period. Action potentials don't occur in neurons alone. They also occur in myocytes or muscle cells. We'll see that in play in the muscle chapter. Eventually, an action potential is going to reach the end of the axon or the axon terminal. And that signal is going to need to jump from one neuron to the next, or from a neuron to a different target cell. Maybe it's a muscle cell or maybe it's a gland. The space between the signaling neuron and its target is called a synapse. So again, the synapse is the junction between the neuron and the target cell. A synapse can be between two neurons. A synapse can be between a neuron and a myocyte or muscle cell. It can also be between a neuron and a gland cell. The process of an action potential changes when it gets to the end of the axon. Instead of triggering the opening of sodium channels, voltage-gated calcium channels are open, and calcium flows into the cell. This influx of calcium will trigger the release of special molecules called neurotransmitters. These neurotransmitters are chemicals that flow out of the axon terminal, travel across the synapse, and then reach the target cell. Here we have a picture of a synapse. In this picture, we can see the signaling axon coming from the top down towards the bottom. This bulbous area here is the axon terminal, and down below is the target cell, which is going to receive the signal. So an action potential is traveling down the axon. That's step one here. Remember that sodium ions are flowing into the axon, changing the voltage. Here you can see in blue, these little circles are sodium ions, and they are rushing into the neuron here. That change in voltage is going to cause the opening of calcium channels, and now calcium will flow into the cell. This influx of calcium triggers these vesicles to release their contents. These are called synaptic vesicles. They're little membrane bubbles that are loaded with neurotransmitters. They just hang out here. they wait and wait until calcium flows in. And when that happens, these bubbles are going to fuse with the membrane of the axon terminal. When those bubbles fuse, they release their contents, the neurotransmitters, out into the synapse. Those molecules diffuse across the synaptic cleft and reach the target cell. At the target cell, these neurotransmitters bind to chemically gated ion channels. Do you remember those from one of our previous slides? They were the other type of gated ion channel. And they open, instead of responding to a change in voltage, they respond when a chemical molecule binds to them. In this case, these are chemically gated sodium channels. And when a neurotransmitter binds to them, they open and allow sodium to rush in and flow into the target cell. If enough of these chemically gated ion channels are opened and enough sodium rushes in, you will get a change in voltage above the threshold, and now voltage-gated sodium channels can open and an action potential will resume in the target cell. And that kind of completes the whole process of an action potential and one neuron signaling a target cell. Here's an electron micrograph of a real synapse. Here to the upper right is the signaling cell. So this is the axon terminal of a neuron. The axon is to the upper right. The kind of bulging terminal is here in the middle. And you can see inside of the terminal are all these little bubbles or circles. Those were the synaptic vesicles. They are loaded with neurotransmitters. So they just hang out and wait for an action potential to tell them to fuse with the membrane and release their neurotransmitters. Down to the lower left is the target cell. This looks to me like the dendrites of a target neuron. Remember, dendrites are the structures that receive signals from other cells. So here this dendrite is just waiting to receive neurotransmitters from the signaling cell. When that happens, that'll help trigger an action potential in the target cell. Action potentials aren't the only type of signal that travels down a neuron. There's also graded potentials. Graded potentials are different from action potentials in several ways. First, they occur on the dendrites and cell body of neurons. Secondly, they involve chemically gated ion channels instead of voltage-gated ion channels. Graded potentials can vary in strength. They can be weak or strong, instead of an all or nothing process like we saw in an action potential. In a graded potential, many inputs are summed, hence the name graded. So there are inhibitory inputs and excitatory inputs, meaning there are signals that help trigger a graded potential, and there's those that help inhibit graded potentials. Inhibitory signals are called inhibitory postsynaptic potentials, or IPSPs, kind of a long name, but they're caused by inhibitory neurotransmitters. These neurotransmitters will bind to chemically gated ion channels that cause an ion to flow into the neuron that hyperpolarizes the neuron, makes it even more negative, reducing the likelihood of an action potential occurring. An example of an inhibitory neurotransmitter is GABA. This is the neurotransmitter that's produced when you drink too much alcohol. Graded potentials can also be excitatory. These are called excitatory postsynaptic potentials, or EPSPs. These are caused by excitatory neurotransmitters. They help lead to depolarization. So when these neurotransmitters bind to their channels, they allow ions to flow in that will cause the inside to become more positive. What's the point of a graded potential? That's to trigger an action potential or inhibit an action potential. And it's the sum of all these inhibitory signals and excitatory signals that will ultimately determine if an action potential can occur. So if the excitatory signals outweigh the inhibitory signals and enough positive ions flow in, that'll trigger those voltage-gated sodium channels to open in the initial segment of an axon, and then an action potential occurs. But if the excitatory signals are overwhelmed by inhibitory signals, then the inside might become hyperpolarized, and this causes a blockage or will stop action potentials from occurring. This slide here depicts the process of a graded potential, which may or may not lead to an action potential. The two signaling cells are colored purple, and the target neuron is yellow. The purple cell to the left is sending out excitatory signals, or EPSPs. These are going to travel the synapse, bind to chemically gated ion channels, and they cause an influx of positive ions into the neuron. In this case, it happens to be sodium. Here's another signaling cell from the upper right. It's sending out an inhibitory signal. You can see it's an IPSP, and that the ion that's triggered to flow in is a negative ion. It's going to lead to hyperpolarization. In this case, it's chloride. But overall, there's far more sodiums rushing in than there is chloride ions. So overall, the inside is going to be more positive. And then if enough of these positive ions overwhelm the negative ions, at the initial segment in the axon of this neuron, those voltage-gated sodium channels open and an action potential can occur. Conversely, if the inhibitory signals were to overwhelm the excitatory signals, this would stop an action potential from occurring. So a graded potential is the process of summing up all the negative and positive ions that flow into the cell and trigger or block an action potential in response. And that concludes part 2 of our voiceover lecture on Chapter 7, the nervous system. Stay tuned for parts 3 and 4.